Hello everyone, and welcome to this audio podcast. This audio podcast as part of the LabZtorm and Squal. This audio podcast is part of the Sepulchre and Storm and Bostics. This audio podcast as part of the Shipmo Squad, and this audio podcast as part of a Storm Robotics production. So through this audio podcast, a little bit in around coping and coping on experimental immune function. So whether there is a means of whether there is a means of habituation is important since this determines whether there is a coping response, whether there is a coping response. Healthcare professionals, for example, cannot just look at aspects in around challenging themselves, in around aspects of challenging themselves in context of young people, and sort of making and then sort of making assumptions about the about sort of whether, for example, a sort of particular challenge prompts a stressful prompts a sort of a stressful prompts a sort of a stressful sort of a sort of stressful basis. Clinical anxiety, is associated with increased circulating neutrophils and fewer NK cells or natural killer cells, T and B lymphocytes, T helper cell and suppressor cytotoxic T cells. Anxiety is also associated with reduced NK cell natural killer cell activity and lymphocytes and lymph sorry lymph and lymphoproliferative responses to to mitogen or cell division stimulation. In one particular investigation, a psychological strain had induced was induced by where was induced wherein volunteers were confronted with a sort of a sort of with, for example, a potentially potentially sort of challenging interpersonal situation. And in this works, participants were asked to solve a complex challenge, and subsequently participants were requested to explain the solution that they came up with to other participants, and so with each other. And the experimental situation induced mild psychological strain, as documented by documented as part of as documented as sort of in line to both in line to sort of in line to their response, the responses, and their responses as they went as and when they were explaining the solution the sort of solution that they used amongst each other in the amongst the experimental cohort. And this was compared with a control cohort as well. Peripheral blood was also drawn by the use of an indwelling catheter just before, directly, and 15 minutes after, and 30 minutes after the stress challenge. Then, in as part of the in as part of this in part of the analysis was where the number of lymphocytes leukocytes, lymphocytes, monocytes, T cells, subsets, natural killer cells or NK cells, and B cells were determined. The control subjects, control participants were assigned to a neutral sort of relaxing condition. A neutral condition to control the effects of the blood sampling procedure, for example. And in the study, in the investigation, the investigators concluded that the effects of a short testing mild psychological stressor are mainly restricted to and in around the natural killer cell population or an NK cell population. Additionally, in another investigation, the effects of acute psychological stress and exhaustive physical activity on the expression of density of adhesion molecules and intracellular adhesion molecules, or ICAM1, on monocytes and granulocytes and lymphocytes were again where these particular cell types were investigated through an acute psychological stress investigation. And in the investigation, participants were seated, and a catheter was inserted into the forearm vein, and this and then rested for 30 minutes. In a fixed order, participants performed a 15-minute speaking task and a 15 to 18 minute physical activity task. And there was a duration between there was a resting duration between the completion of each task and the start of the next task. So in this investigation, the data indicated or informed that both psychological stress and physical activity have significant effects on cellular expression of adhesion molecules on circulating leukocytes, on circulating leukocytes. So that draws this audio podcast to a close. This audio podcast as part of the LabZtorm and Squal. This audio podcast as a part of the Sepulchre and Storm and Bostics. This audio podcast as part of the Ship Mode Squad, and this audio podcast as part of a Storm and Bostics production.